Hey Biology, so today we're going to go ahead and talk about controlling cell growth, and we'll talk about cell differentiation. That's chapter 10.3 and 10.4. So let's first talk about cell division and how frequently cells divide. So we have some cells in our body, like our skin cells, in which, like we talked about in class, basically most of the dust we see in the air is our dead skin cells. So we lose skin cells often. So our cells have to divide at least once an hour. Some cells will divide occasionally. So our liver cells, they divide about once a year. And then some cells don't divide once they form, our nerve cells, which once you have nerve damage to some things, that's the reason why that can't be repaired on its own. So we have things that regulate the cell cycle. We have internal regulators and we have external regulators. So external regulators basically are proteins that will respond to the events outside of the cells. So they're just signals that tell the cell to speed up or slow down the cell cycle. And an example of that would be growth factors. So growth factors basically stimulate cells to divide, and that's important during wound healing and embryo development. So whenever we get injured, obviously we're going to start to bleed, and we bleed because our cells are responding to an external signal. So cell division genes in the cells are near the injury turn on to heal and replace damaged missing cells, and turn off when the repair has been done. We also see this in example when cells receive signals from neighbors. So basically we have cells right here that are in clusters, and they're going to continue to grow until they all touch each other. And then when they touch each other, they stop dividing. So that is contact inhibition. Well, if we were to wipe that plate clean, our cells would then continue to divide, like you see in this cell here. And they would divide and divide until, again, they touch each other, and then they would turn off. We also then have internal regulators. So that's when proteins respond to events inside a cell. So the cell cycle proceeds only if certain processes have happened. So a cell can't enter mitosis until all of the chromosomes have been copied in S phase. So in the early 1980s, scientists discovered a protein in dividing cells that caused a meiotic spindle to form in non-dividing cells. So basically, if they were to inject this protein into a non-dividing cell, the cell would then start the process of mitosis. Levels of this protein rose and fell with the cell cycle, so it was named cyclin because it seemed to control the cell cycle. We actually have found a whole family of cyclins, and we've discovered that they regulate the timing of the cell cycle in eukaryotic cells. So most cells divide up to 20 to 50 times in a culture, then they stop, age, and die. So cells only divide a certain number of times. Cancer cells are considered immortal. So HeLa cells from a tumor removed from a woman, Henrietta Lacks, in 1951 are still reproducing in a culture. So her cells don't have that timeline. Dolly, for an example, is can be shown as a way that cells actually do grow, stop, age, and die. So Dolly was the cloned sheep. She actually died of old age at 6.5. Well, she died of old age at 6.5 because they took a stem cell from the original animal that she was basically cloned from, and those cells in the DNA in her cells were old to a certain point. Well, because they were so many years old, that's how old Dolly was, or that's how old all of Dolly's cells were when she started to grow and divide as well, or at least that's how old all of her DNA was. So cancer is complicated and can have many causes, but all cancers have one thing in common: they have lost control over their cell cycle. Many cancer cells have damaged or defective tumor suppressor gene called p53. And p53 basically signals DNA to repair enzymes, holds cell at that G1 checkpoint, and starts apoptosis of damaged cells. So apoptosis just means cell suicide. So if the cell finds something wrong within it, it'll basically destroy itself to save all of the other cells. So cancer cells have no contact inhibition. They don't stop dividing when they touch nearby cells. They just keep growing and growing. and that's what makes basically the tumor. Now, tumors can be considered, and we've talked about this, or you read about it in your cancer book, benign or malignant. Now, benign tumors, that's what you want to hear if you have a tumor. They're just abnormal cells that stay at their original site, so they don't move, and they can, for the most part, be removed easily by surgery. Malignant tumors, those are the scary tumors. Those are the cells that actually leave the original site, and they basically can be carried by the blood and lymph systems to other tissues, and that basically starts then more tumors called metastasis. They basically impair functions of organs throughout the body. Those are your scary tumors, malignant. Benign are the ones that, no matter what, tumor's scary, but those are easy to remove. So cancer cells, they don't respond to control signals. They lose contact inhibition, so even if they're touching other cells, they'll continue growing. And transformation process that changes a normal cell into a cancer cell, so they can transform normal ones into cancer cells. So again, they don't stop dividing. Like a car with no brakes, metastasis basically can spread to new places. That's what that means. So our malignant tumors, we would consider metastasis, meaning that they can spread. Carcinogens are actually substances that damage DNA and cause cancer. So cancer does run through gene lines, so it can be hereditary. However, carcinogens are things that even damage DNA more and can give you a higher probability that you'll get cancer. So cigarette smoke or chew, UV radiation, chemicals, pollution, genetics, viruses. So traditional treatments for cancer, well, we know that there's chemotherapy with toxic drugs or high energy radiation and chemotherapy. That's traditional methods. There are different holistic methods, but usually what hurts cancer patients most is the high radiation and the chemotherapy, because radiation in your body makes people sick, or the high radiation, it can damage all the cells living around the cancer cells as well. The chemotherapy, basically what you take in with the toxic drugs, that can also make you sick, but it helps to kill the rapidly dividing cells. Okay, so how are cells then different? So one of the most important questions in biology is how all of the specialized differentiated cell types in the body are formed just from a single cell. So we start off with the fertilized egg. So you have your mom's DNA and your dad's DNA. Well, how does that one cell turn into many more cells? Well, biologists say that such a cell is called topipotent, and that means literally to be able to do everything. So our embryonic stem cells we consider topipotent. Only the fertilized egg and the cells produced by the first few cell divisions of the embryonic development are truly topipotent. So that first fertilized egg and then the few cells after that. So about 4 days of development, a human embryo forms into a blastocyst, and this is basically a hollow ball of cells with a cluster of cells on the inside. The cells on the inner mass are said to be pluripotent, so not topipotent. So topipotent again can turn into anything. Pluripotent means that they have the ability to develop into many, but not all body types or body cell types. Stem cells are basically the unspecialized cells from which differentiated cells develop. There are two types of stem cells. There's the embryonic, which is the big controversy is over, and then there's the adult stem cells. Embryonic stem cells are found in the inner cell mass of the cell embryo, and embryonic stem cells are considered to be pluripotent, so the ones found in the inner cell mass. Topipotent ones are those first ones. Researchers have grown stem cells isolated from human embryos in cultures. Their experiments confirm that embryonic stem cells have the capacity to produce most cell types in the human body. So we also have adult stem cells. So your body has stem cells in it as well. So adult organisms contain many types of stem cells. We call these adult stem cells multipotent. So they can produce many types of differentiated cells, just not as many as the pluripotent or as many as the topipotent. So adult stem cells all give an organ or tissue cells are given organ or tissue typically produced by the types of cells that are unique to that. So potential benefits. Stem cell research may lead to new ways to repair cellular damage that result from heart attack, stroke, and spinal cord. We watched a movie where this guy, he's suffering from heart disease, and they inject stem cells into his heart. So this guy, before he had this injection, could barely walk. Afterwards, he could actually do layups. Okay, so then we have some ethical issues. So what we're talking about. Most techniques for harvesting or gathering embryonic cells cause destruction to the embryo. So something that could eventually be life, or embryos are sometimes considered life. It depends on the type of person you are and what your beliefs are. So government funding of embryo stem cell research is an important political issue. Obviously, we saw Bush, we saw Obama. They had very different views on how it should be funded. There's groups that seek to protect embryos. They oppose such research and they consider it unethical. Other groups support the research and think it's essential to saving human lives. So that's going to be it for chapter 10.3 and 10.4 notes. Don't worry about getting anything down in your notebook.